Today we will try to understand the structure of a DNA molecule. DNA or deoxyribonucleic acid is one of the four major types of macromolecules present in our bodies, the other three being carbohydrates, lipids, and proteins. All these macromolecules are polymeric in nature, meaning that they are composed of small molecules called monomers, which are joined together to form long chains of large molecules known as polymers. The monomeric subunit of a DNA molecule is known as a nucleotide. It consists of a five-cornered ring structure known as a deoxyribose sugar attached to a nitrogenous base at its carbon 1 and a phosphate group at its carbon 5. It is known as deoxyribose sugar because it's missing one oxygen at its carbon 2. The phosphate group at each nucleotide forms a covalent bond with the hydroxyl group at carbon 3 of the adjacent nucleotide to form a long chain. There is a free phosphate group at carbon 5 known as the 5' end of the polynucleotide chain and a free hydroxyl group at carbon 3 at the other end known as the 3' end. The phosphate group and the deoxyribose sugar form the backbone of the DNA molecules with the nitrogenous bases directed towards the sides. A DNA molecule contains two different types of nitrogenous bases, purines and the pyrimidines. Purines are the double-ring nitrogenous bases and their examples include adenine and guanine. Pyrimidines, on the other hand, are the single-ring nitrogenous bases and their examples include thymine and cytosine. A complete DNA molecule appears as a double helical structure with two polynucleotide chains running along each other in the anti-parallel direction. One chain runs from 5' to 3' end, while the other chain runs from 3' to 5' end. Both these chains are held together by hydrogen bonds present between the nitrogenous bases of each chain. The pairing between the nitrogenous bases is highly specific. Adenine only binds with thymine by the help of two hydrogen bonds, while cytosine only pairs with guanine by the help of three hydrogen bonds. This hydrogen bonding between the nitrogenous bases stabilizes the DNA molecule to a great extent. The double helical molecule has major and minor grooves with about 10 base pairs in each turn.